I welcome you one and all for today's section.
Well, thank. Well, as per the schedule, shall we get the introduction of today's speaker? So before going to the speaker's introduction, our principal sir has been hectic with some of the official works, so he couldn't able to join the session. And sir has to convey his wishes and regards to the delegates as well as for the speakers. And to this session, before introducing the speaker, let me say a few instruction to the delegates. Kindly please mute yourself as well as please stop sharing your screens. And the question session will be with the end of the session. And in meanwhile, if you have any doubts, you can please post in the chat box. Well, so let me introduce the speaker today's speakers first. It's an immense pleasure for me to introduce my beloved senior, Dr. N. Jawahar. Dr. N. Jawahar earned his B Pharm from Arulmigu Kalaslingam College of Pharmacy in 1999 and pursued his M Pharm from KM College of Pharmacy 2002 and awarded PhD from JSS University in 2013, Mysore. And experience includes both teaching and research of 18 years. Received several awards, including Career Award for Young Teachers, CAYT 2012, AICT India, and Tony B. Academic Travel Award, SLAS 2013 USA, and Best Industrial Pharmacy for Poster Award, FIP 2010 Portugal, and Best Presentation Award, FAPA 2018 Manila, Philippines. Travel grant received from various funding agencies such as DST, CSIR, ICMR, SLAP, and FIP for attending various conferences. Dr. N. Jawahar has published 65 articles in peer-reviewed index journals with impact factor ranging from 1 to 6 and presented 30 research presentations in national and international conferences. He also authored 2 books by Lambert Academic Publishing, Germany, and 4 book chapters for Apple Press, USA, and Rivers Publishers, UK. He has filed 2 Indian patents, application number 2194, and he involved as a resource person in national level seminars and delivered 20 lectures in various universities across Tamil Nadu. He has guided 50 PhD students and guiding currently 5 PhD scholars. The major research area of Dr. N. Jawahar is formulation and in vitro evaluation of nano drug delivery system for improving the bioavailability or drug targeting to specific site tissues, organs. He is a lifetime member of various professional bodies like IPA, APTI, APP, AAPS, and SPDS. He has been recognized as a reviewer and editorial board member of various international or national indexed journals. So, with this brief introduction, I would like to call Dr. N. Jawahar to present his talk today. Over to Jawahar, sir, please.
Thank you, Dr. Ramkant, for the nice introduction. And it's my privilege that Ramkant is my junior. And now he is holding the position of professor and head of Kabam College of Pharmacy, organizing this particular webinar. Now, I'm just sharing the slide. Is it visible? Hello, am I audible?
Yes, sir. You're audible, sir. Slides.
But screen is not shared.
Is it when you're audible? Right now?
No, sir. No, sir.
I didn't, I mean, get this screen to share. Yeah. Okay. Yeah, this is the slide. Hope now it is shared.
It will
Hello.
present, but it's showing presenting. Not at.
I think
presented.
it will take a moment, it seems.
Yes, yes.
Just wait and let
Yeah,
us know.
It came itself. Shared.
Yeah. Okay. So,
Yes, sir. Okay.
yeah, fine. So, very good morning to one and all, respected teachers and students. So today my topic, what I'm going to deliver is solid lipid nanoparticles, a promising career in drug delivery system. Though the topic is, mentioned as solid lipid nanoparticles, I'm not only going to deliver about only SLN, I'm also going to deliver some aspects of NLC. See, NLC is nothing but the modified form of SLN. So that's why I didn't mention SLN bar NLC. Okay, so that's the reason, but we will discuss both simultaneously together. Coming to the introduction, you know, nowadays there are a lot of drug development and drug discovery programs are made, and there are so many new drugs are developed from such kind of program, especially with combinatorial chemistry like in silico tools using computer-aided drug designing and all. So they are coming with a lot of novel moieties, new molecular entities, and you know, the developing a new drug is not only sufficient to ensure the progress in drug therapy, because there are many newly developed compounds, newly developed molecules, which are showing very nice, binding efficacy with the particular receptor for many diseases, are showing very disappointing results in vivo. You all know that. So the main reasons for such kind of, incidents is nothing but the therapy. We can say it's a therapy failure. So the reason for that therapy failures are insufficient drug concentration. So though it's showing a very good what is called binding efficacy or activity in vitro level, but when it comes to clinical trials, it will not show that much progress in the efficacy. The main reason is insufficient drug concentration to the site of action. Okay. Second one is drug distribution to other tissues. Okay. For example, especially I can say an example for anti-cancer drugs. So anti-cancer drugs supposed to go for the cancer cells, but in conventional drug delivery system, it's not only going for the cancer cells. It also what is called accumulated in normal cells and what is called produce some sort of adverse effect or toxic effect to the normal cells. And for cancer cells, it will go with lesser concentration. So that leads to therapy failure. Then poor drug solubility. Yeah, this is the first and foremost issue in the current scenario, you know, because most of the drug molecules developed in are available in the market comes under the category of BCS class 2 or BCS class 4, you know very well. So BCS class 2 is nothing but poorly soluble and highly permeable, whereas BCS class 4 is both poorly soluble and poorly permeable. So those drugs will not able to achieve the better bioavailability in terms of solubility or permeability. So which in turn leads to poor pharmacokinetics in patients, leads to therapy failure. The next one is high fluctuation in plasma levels. Yeah, some of the drugs in the conventional drug delivery system or conventional dosage form may increase, sudden increase of the concentration in the blood. Quick Cmax and quick elimination, which in turn what is called frequent doses are required. If with more doses may leads to some sort of side effects or adverse drug reaction. So these are some of the reasons for therapy failure. So as I told, a number of molecular entities selected for full-scale development based on their safety and pharmacological data suffer from undesirable physicochemical and biopharmaceutical properties, which in turn poor pharmacokinetics and distribution. That's what. So even nowadays also many molecules just developed, developed, and it's given for the trial. I mean, even in the COVID situation, you can see there are people are talking about many vaccines, drugs. Okay, working in vitro might be, or what is called in even preclinical studies also it shows some good effect. But when it comes to the human trials, clinical trials, it will not work out. Why? Because of these reasons. The particular molecule having very poor physicochemical property, which in turn poor biopharmaceutical properties, which leads to poor pharmacokinetics. Okay, so in this context, a potential role of drug delivery system has to be explored in order to modify the physicochemical and biopharmaceutical properties of such drugs in order to improve their bioavailability and reduce toxicity. So this is the reason. Here, the drug delivery scientist, the role is exactly this one. See, the many molecules which are discovered from various tools may have a lot of issues, physicochemical issues, bioavailability issues, pharmacokinetic issues. So all those issues can be overcome by converting them into a suitable delivery system. So the delivery system will alter the nature of, physicochemical nature of the drug, but it will not affect the drug-like property. So thereby it will improve the bioavailability and reduce the toxicity. So one of such drug delivery system is nano drug delivery system. You all know. So nano drug delivery system is a promising approach in order to obtain desirable drug-like properties. That's what I told in the previous slide also. Here the drug-like properties will not change. Just we are altering the physicochemical and biopharmaceutical properties of the molecule. Converting any drug into nanoparticle or nano drug delivery system will not alter the drug-like properties. Its nature of the drug will not change, never change. But the physicochemical property of the drug will change. That's the added advantage of the molecule to overcome whatever the issues, what we discussed in the very first slide. No, there are many issues we discussed for the reason that leads to therapy failure. That will be overcome by this kind of drug delivery system. So nano drug delivery system alleviate the unwanted toxicity. That's what, due to non-specific distribution. So yeah, I want to correlate the first slide with this point. One of the point I said, it will bind to a non. It will distribute it to the normal cells. I told one example, cancer, anti-cancer drugs, not only what is called binding or reaching to the cancer cells, it also goes to the normal cells. So that can be avoided. So, when an anti-cancer drug reaching to a normal cell will be harmful for the normal cell, which in turn the normal cell also will get what is called killed. So which in turn a lot of side effects or adverse effects may occur to the patient. So that will be alleviated by what is called site specific or organ specific or to the particular cells specific. We can target the drug from the site of administration, okay, to the site of action. And also it improves the patient compliance and provide favorable clinical outcomes. Obviously, so you see when a patient is avoided with side effects or something, means he feel better, and he will take the medicines properly, and the therapeutic effect also will good. So since the last decade, from 1990 onwards, the nano drug delivery has taken a new dimension with the increasing application of nanoparticles as a carrier for delivering of poorly lipophilic drugs. Not only it's limited to lipophilic drugs, even sometimes we can incorporate some sort of hydrophilic drugs also to the nano drug delivery system. So the most investigated nano drug delivery system for the past 2 decades are polymeric nanoparticles, which are using with both synthetic polymers as well as natural polymers, and lipid nanoparticles. It's mainly SLN, you know what is, it's nothing but solid lipid nanoparticles, and the modified solid lipid nanoparticles, I can say, NLC. Though it's both are comes in the same generation, only there is a modification change, structural modification change. We can differentiate SLN and NLC. Rest of the property and everything are almost similar. And the third one is nano suspensions and nano emulsions. So coming to the definition of solid lipid nanoparticles as well, nanostructure lipid carriers. Okay, so both SLN or NLC are tiny colloidal carriers composed of biocompatible or biodegradable lipid matrix that is solid at body temperature. Not only body temperature, even at ambient temperature, it will be solid. And it exhibits the size range between 10 to 1000 nanometer. But in, you know, in many papers, they may, what is called, vary with the size range. Some of them will say it's 40 to 1000. Some of them say it's 100 to 1000. So it's all depends upon the, what is called, the application, what we want to use the nanoparticles. So there the sizes matters. But in general, all the pharmaceutical nanoparticles, not only the SLN, even polymeric nanoparticles, I mean, lie between the size of 10 to 1000 nanometer. You know, it consists of spherical lipid nanoparticles in the nanometer range, which are dispersed in water or in aqueous surfactant solution. Coming to the NLC, so NLC matrix is composed of mixture of spatially different lipid molecules. Normally, the mixture of solid and liquid lipid, which makes more imperfection in the lipid matrix in order to accommodate more drug molecules than SLN. That's the difference between NLC and SLN. See, NLC, we are using only solid lipid, whereas, SLN, whereas in NLC, we are using both solid lipid as well as and liquid lipid. So what will happen in SLN? The structure of SLN will be the perfect structure. I mean, the matrix structure will be perfect, whereas in NLC it is imperfect. So because of that imperfection, the more amount of the drug will be accommodated or loaded, and moreover, the drug expulsion also is very less in NLC than SLN. So it is expected that the drug loading capacity will be enhanced, as well as drug expulsion during storage will be minimized due to the imperfect crystal lattice, and drug release profile can be easily modulated by varying the lipid matrix composition. What happened to the SLN upon long-term storage means, because of the perfect structure, it will start, I mean, it will be like perfect crystalline structure. Generally, you know what will happen to the crystalline structure during the storage condition. Slowly it will degrade and it will convert it into amorphous. But whereas NLC, you know, already it is an imperfect structure. It's like you know, semi amorphous. I cannot say 100%. It is also a crystal only, but you know, because of the imperfections in the lattice, it will take a long time to convert them into amorphous completely. So NLC is much stable than SLN. Though SLN is having the hard matrix, many people may have the question because of we are using solid lipids, no, in SLN. So obviously it will produce a hard matrix. Yeah, obviously matrix wise SLN is very hard, whereas NLC is very less. But you know, this is the reason how it will differs to increase the stability. So coming to the advantages of SLN or NLC, so it can have the ability to entrap both lipophilic as well as hydrophilic drugs. Okay, anything is possible. Then no biotoxicity, because it is completely biodegraded. People may ask, I mean, if in that case there is no toxicity for SLN or NLC. Yeah. As far as literature is concerned, and I mean studies are concerned, so far no report has been provide with the toxicity issues of SLN or NLC, because we are using the physiologically accepted lipids for the formulation. So obviously, how we are taking the fat in terms of anything, meat or oil in our day-to-day food, how it will not produce much toxic, if it is taken in a right amount for the right time. Okay, so similarly, this SLN or NLC also, we are using the lipids or the fat materials which are more biocompatible. So completely it will be biodegradable in the body. So which in turn it will not produce any biotoxicity. But sometime there is a possibility. Why? Because of the excipient what we are using for the method of preparation, or I can say the solvent what we are using for the preparation. So all those are some minor reasons. So that also we can overcome by sensibly by using grass like excipients. That means as per the FDA, generally regarded as safe category. There are excipients classifications are there. If you just go to the US FDA website, they have classified the solvents, classified the excipients under the category of grass, generally recognized or regarded as safe. So if you use such kind of excipients, then obviously that also will be avoided. And the protection of liable compounds against chemical degradation, because of its steric stabilization, most of the time it will not get what is called degraded, okay, in the physiological system. I mean, it will not get degraded by the enzymes. Generally lipids will be degraded by lipase or co-lipase enzyme, but when we stabilized with some surfactant material, so that we can avoid. But if you want, that in turn, I mean, the drug particles will what is called release very slowly. If you want quick release, you can use some sort of instead of surfactant, you can use some other coating which will attract the lipase or co-lipase enzyme, which in turn the drug will be degraded quickly and it will be released quickly. So that's all the tricky things. Depends upon the application what you want. You can use such kind of things. So the general, apart from that, enzyme degradation, even pH, nothing will be affect the molecule because of its hard matrix. I mean, the drug is incorporated or entrapped into the hard matrix. Then you can have plenty of routes of administration. It will be used in oral, it will be used in topical, it will be used in parenteral, it will be used in ocular. Sterilization is possible. Yeah, because of its high melting point, most of the lipids are able to withstand the autoclaving methods and all. Especially when the SLN or NLC used for parenteral drug delivery system, it will not get degraded or denatured during sterilization. Especially we can go for cold sterilization rather than autoclaving method. You all know very well what is cold sterilization. It's nothing but the filtration. Why? Because of the size. Since it's a nano size, it can easily pass through the membrane, which are having very less size. 0.3 micrometer or something. So thereby the bacteria kind of things and all will be get retained on the membrane, and only the pure nanoparticles will be collected as a filtrate and may be used for parenteral application. Then drug targeting. Yeah, this is another advantage of SLN and NLC. You have plenty of choice to target to different tissues, cells, or organs, or receptors by using suitable ligand material or doing some surface modification with different kind of things. Even you can add with some antibody, anything you can do. A lot of manipulation you can do with the surface. Then ease of commercialization and scale up for large scale production. Yeah, that's what, because the cost is very less when compared to the other nanoparticles. Lipid cost, everything is very easy, and scale up also very easy. And this is a good delivery system for commercialization. Then regulatory acceptance of excipients, as I told in the earlier, so it will be easily accepted. All the lipids are accepted well by all the regulatory bodies, including US FDA. Then the lymphatic transport. This is the one of the unique advantage or unique feature of this particular SLN or NLC. You know why? Because most of the drugs which are given in the oral route will taken up by the portal vein, go to the liver, and then metabolize, then intact drug will enter into the blood circulation for the bioavailability. All those things you know very well. But you know, here we can bypass or circumvent the first pass metabolism by avoiding the uptake of, drug entering into the portal vein by this lymphatic route of administration. That I will discuss in the later slides with some animation. So by the time you may understand very well. Then these are the comparison of SLN or NLC with the other colloidal carrier right now, what is called worked by many people. I compared the SLN and NLC with liposome. You know, liposome is the first lipid-based drug delivery system. Okay, in 1960s, people are tried with the liposomes, and it's the what I can say in lipid drug delivery system. This played, major time as a unique delivery system till SLN and NLC replaced the liposome. Till that time, liposome was the only lipid-based drug delivery system existed in the market, and there are many works performed with liposomes, but because of some reasons, like its stability, it's a vesicular drug delivery system, leaky in nature, not having the good stability, as well as the cost also matter in the liposomes. Cholesterol and other things and all used. So that will be the reason it will be outdated from the research as well as market nowadays. Then nano suspension, it's another what is called simple as well as the very effective delivery system. Then polymeric nanoparticles and the emulsion, of course, it's a coarse one. So you see, we have compared the various properties of these colloidal carriers, parental delivery, oral delivery, ability to deliver both hydrophilic and lipophilic drugs, physical stability, biological stability, biocompatibility, ease of sterilization, drug targeting, drug loading, ease of commercialization, ability to deliver biotechnological therapeutics like gene therapy or something, then regulatory acceptance. So if you see the comparison, everything is possible, everything is good with SLN and NLC compared to any other colloidal drug delivery system, which are also having possibility of most of the things, but lack with one or other property. So since it's a complete what is called delivery system which covers maximum advantage than the other colloidal drug delivery system. So here I can show some of the lipids which are used for the preparation of SLN and NLC. So it may be useful for you people to try with some sort of preparation with SLN and NLC. So most of the time we can use triglycerides. The partition coefficient value of triglycerin is good, so the solubility of the lipid will be more. So you can accommodate more amount of the lipids in those particles than the other kind of lipids like monoglycerides or fatty acids or waxes or something. But it may what is called vary time to time, depends upon the surfactants. That's nothing but the emulsifiers what we are using and the ratio of emulsifier and lipid. All those things are tricky things that you can understand by doing some sort of optimization during your method of preparation, right? So here I can show some of the liquid lipid examples: soya bean, oleic acid, etc. Then waxes, okay. Then fatty acids, also stearic acid, also we can use. Why this table I have given means many of the what is called lipids are available in your lab itself. Okay, triglycerides may be little costly comparatively, but whereas the other category of lipids are available in your regular lab, so it's very easy to start with this SLN and NLC in your lab. Emulsifier also, again, lipophilic emulsifiers and hydrophilic emulsifier, like I have categorized. So like lipoid, lecithin, phospholycoline, some of the lipophilic emulsifiers, then polaxamine, polysorbates, Okay, these are some of the hydrophilic. So these kind of emulsifiers or surfactants also available. So based on the need, you can use what is called any surfactants which can emulsify much better. So generally, why we are using emulsifier means emulsifier will give the stability, steric stability of your formulation. Okay, so that also you have to choose by trial and error based method. So in which it will give good stability, that may be the suitable one. Then coming to the methods of preparation of SLN or NLC, these are the various methods. High pressure homogenization. Usually it has two types: hot homogenization and cold homogenization. Then solvent evaporation, then microemulsion method, solvent diffusion method, spray drying method, supercritical fluid technology. So some of the methods are very hi-fi methods which requires sophisticated instruments, especially the supercritical fluid technology. Spray drying and all, you have to have nano spray dryer. Then homogenizer means you have to have a high pressure homogenizer. But some of the methods are very feasible and simple. You can able to do with some beakers and normal stirrers. Okay, like solvent evaporation, microemulsion technique, and solvent diffusion technique. High pressure homogenization technique. Usually we have to keep 10 degree more than that of the melting point of a particular liquid. So by the time, and upon high, due to the high pressure, there is a possibility of heat generation, which in turn sometimes, the denature of lipid may occur. So that's why generally I will not prefer with high pressure. So there are papers available. There are people are doing. There are places they are doing with high pressure homogenization. But as far as mine is concerned, it's having a lot of issues in developing the formulation. Okay, so now I am going to discuss with two simple techniques which are very feasible in your lab. And the first and foremost one is microemulsion technique. It's an ultimate method. Why I am telling means, in this we are not using any organic solvent, just we are using the lipid, water, and surfactant. That's all. Nothing else we are going to use, and it's organic solvent free method or formulation. So it's much better for the regulatory acceptance than the other method. Even next method I am going to discuss with solvent evaporation technique. In that we are using some solvent like organic solvent like acetone or something. Even though we are evaporating or removing the solvents, there is a possibility of residues. Even sometimes those residues may do some sort of toxicity with genomic level. Since the particles are nano, it may have some sort of genotoxicity if the size is very small. So by the time that residues will do some sort of manipulation in the gene. So, okay, the first technique is microemulsion technique. It's a very simple technique. So just you can take the lipid and your lipophilic surfactant. That should be melted or heated at 70 degrees Celsius. At the same temperature, you have to heat the water along with your aqueous surfactant like polaxomas, okay, or fluoronic F68 or 127 or something like. Okay, both the things. The melted lipid will be added into the hot aqueous surfactant with stirring. Okay, the stirring speed, stirring time may change. You can optimize. Depends.
138 இருக்கானுங்க.
Excuse me. So that you can decide. You want what size of the particle if you want. Accordingly, you can set your stirring speed and stirring time, and the concentration of lipophilic surfactant and lipid. That ratio also will matters for the size. Depends upon the size or something. So then after that, it will form a warm microemulsion. Then the microemulsion will be added into ice cold water. Then it will get the particles will get precipitated. Then the precipitated one will be centrifuged. Then the supernatant will be decanted, and the concentrated one will be taken for freeze drying. So upon freeze drying, you will get the free flowing powder which are physically stable. You know, here I would like to tell one more point that, the physical stability of NLC and SLN, both are same in terms of lyophilized form. Okay, nothing is different. If it is in the aqueous solution, or what is called, it's dispersed in organic solvent or aqueous solution, that time only the stability issue will be vary from SLN and NLC. But in the lyophilized form or freeze-dried form, the physical stability is much better, equally in SLN and NLC. So during the freeze drying, you can use the maximum amount of 1 to 2% of cryoprotectants or cryoprotective agents, like mannitol, sucrose, okay, kind of some kind of carbohydrates, which are all used as a cryoprotectant, which will prevent the agglomeration or oswald whitening during the freeze drying process. So the stability of the nanoparticles upon freeze drying will be improved. Even the lipophilic surfactant, you can use the concentration between 1 to 5%. Okay, that's the optimum range. And lipids, you can use 0.1 to 30%. Okay, that concentration you can take for your formulation. So coming to the solvent evaporation technique. So this is a simple technique. That's why I'm again discussing. So in here also, instead of only small thing is instead of water, we are using organic solvent, okay, which are partially miscible with or dissolved with water. We can use combination of acetone and water or something. Or as such, the organic solvent also we can use cyclohexane, acetone kind of things. Okay, so the same way, lipid and lipophilic surfactant will be melted and added into the organic phase. Okay, then it will be the first it will be added into the organic solvent for miscibility. Instead of melting, we can just miscible. I mean, the lipids will be dissolved, then it will be added into the aqueous phase, then it will be stirred for a long time so that the organic solvent will get evaporated. Then we will do centrifugation and we will go for freeze drying in order to get the free flowing lyophilized form of nanoparticles. So upon completion of formulation, this part is very important for nanodrug delivery system. Till then, very easy, you can do with your lab facilities. But for this particular aspect of SLN, I mean characterization part is very important. We have to go for some sophisticated instrument to determine the various properties of nanoparticles. So the first one is measurement of particle size and zeta potential. So you all know particles and zeta potential play, as well as polydispersity index, PDI, plays a major role of nanoparticles. Okay, so size will determine the targeting, especially for targeting. Some of the barriers will not allow with high molecular weight and the size of the particle is high. So that's why. And polydispersity index again matters with its stability as well as its uniformity, content uniformity. So if the particle size, PDI, polydispersity index, is very less, it's close to 0, means most of the particles are uniform in size. It's called unimodal. Okay, if the particle size close to 1, means it's a polymodal. It's the particles are available with varying in size, different in size. So always uniformity have better advantage than the non-uniform particles. And the last one is zeta potential. That's very important. That is nothing but the surface charge of the particle. So it plays a major role in stability, physical stability. You all know, if your nanoparticles having plus or minus 60 volt, means it's 100% absolutely perfect stable in terms of physical stability. If it falls between plus or minus 30 volts, means it's optimum stability, and plus or minus 20 volt means desirable one. okay, desirable one. So from that surface charge, we can understand the stability of the particles. So those are all determined by these different instruments like photon correlation spectroscopy, laser diffraction, atomic force spectroscopy. Then the measurement of crystallinity. Yeah, this is another aspect which will be measured by differential scanning calorimetry. So here we can what is called have the preliminary idea about the crystalline form or amorphous form of our developed nanoparticles. You know, for example, before formulation, the drug is crystal in nature. After the formulation, if you see the thermal behavior, that how much heat it absorbed or something, there is a variation. That means if the endothermic peak is nil or something, that means it's completely converted into amorphous. Usually in the crystalline form, it may have some sort of melting point, but when it converted into amorphous form, the melting point will be vary. So it will not absorb much heat than the crystalline form. So such kind of idea, which means the drug will be completely soluble into the lipid in the formulation. That's the understanding or interpretation or conclusion. Okay. Then if you want further detailed modification, how it is modified, the lipid is modified or the drug is modified into the lipid, means we can go for X-ray diffraction studies and IR infrared spectroscopy kind of studies and all. And especially when we are doing for some semi-solid dosage forms incorporating SLN and NLC, we can use some rheometry in order to check the viscosity property and all. Then surface morphology, we can go with two. One is SEM, you all know, and another one is TEM, that is transition electron microscope. Ram, excuse me. Hello. I'm too late, or what time I'm supposed to complete? Yeah, fine. Okay then.
Yeah.
Okay then. So the other characterizations are entrapment efficiency, drug content. So in surface morphology, SEM is very less cost than the transmission. So generally, for the purpose of research, we can go for SEM. For the detailed, if you want to know the detailed surface morphology, then only we can go for transmission electron microscopy. Otherwise, not necessary. Then the other properties are in vitro studies and in vivo studies. So coming to the application, as I said, SLN NLC is used for different routes of delivery. The major and more comfortable route, you all know, is oral delivery. So SLN and NLC have been used to enhance the oral bioavailability of several drugs by using this lymphatic transport. Here, these are the steps involved. First, the drug will flow into the GIT, then it accesses the M cells of Peyer's patch, or otherwise called enterocytes. Then it will be entered into the mesenteric limb and followed into the lymphatic vessels, then transport between the lymph and blood. So this is the lymphatic transport mechanism. Just I will show you what is called animation. So drug is flow and get into the M cells of Peyer's patch, then it enter into the lymph vessels, and then finally it will enter into the blood vessels. So thereby it's a what is called direct absorption of drugs from the GIT to the blood by avoiding first pass metabolism, which in turn bioavailability will be enhanced. Okay, so this is the case study. We developed the SLN in the year 2010, and with the drug olanzapine, it's an antipsychotic drug, which is a PCS class 2 drug. In that, we have increased the bioavailability of 4.33-fold than the blind drug, and the possible reasons are direct uptake of nanoparticles through GIT, increased permeability of intestinal membrane, bioadhesion. Yeah, most of the lipid particles are having a bioadhesive nature. And then another thing is we are using the positive charge inducer. So thereby we can what is called change the charge of our particles. We use sterile amine as a positive charge inducer. So thereby the reason for inducing the positive charge is the positive charge particles are well taken by the negative charge intestinal mucous membrane. So thereby it will absorb in more amount and enter into the lymphatic transport So thereby the concentration of the drug available in the blood is increased. And this is the paper we published in the current nanomedicine. Then the similar way we tried with NLC with the same kind of drug, I mean antipsychotic drug olanzapine, and that also shows excellent enhanced bioavailability than the SLN. And that also published in the International Journal of Biological Macromolecules. And even we targeted with SLN to the brain, and we succeeded in that aspect also. So we used polysorbate 80 coated SLN. It will what is called absorb the ApoE lipoprotein in the body. This ApoE lipoprotein will cheat the brain barrier, blood-brain barrier, because it will cross through LDL, low density lipid receptors. So thereby we increase the brain bioavailability. Just I will show you. So the drug will be attached with polysorbate 80. So the polysorbate 80 will absorb the ApoE lipoprotein. So it will mask himself like ApoE lipoprotein, and then it will enter into the LDL receptor of the BBB, and then it crosses the BBB. So thereby we enhance the concentration. So we enhance like 23 folds enhanced brain concentration than the normal drug. The reasons are slow release of the olanzapine, as well as low clearance, as well as this what is called mechanism. We used polysorbate 80. So that also published in the journal called Artificial Cells in Nanomedicine and Biotechnology. Then this SLN is very good in injectable drug delivery, that is parenteral drug delivery, because of these reasons: low excipient loads, low dose, better control of particle size, increased efficiency, capability of sterilization, etc. So you may so these are some of the drugs which are already what is called studied for parenteral drug delivery. And next one, topical delivery. So in topical route also, many of the drugs having poor permeation, so thereby it will not fulfill the purpose. So but SLN and NLC will increase the drug loading capacity, potential of decreased skin irritation, stabilization of sensitive actives, and control is physical and chemical long-term stability is possible with topical. And these are some of the drugs which are studied for topical application. And this is the mechanism. Generally normal what is called conventional creams, gels, only very less particles will enter. So majority of the drugs will retain on the skin surface because of the size. But if you use permeation enhancer, what will happen? It will tear the lipid bilayer. Thereby, not only the drug, apart from the drug, the bacteria, kind of microbial organisms also will enter, which will worsen the case. But if you prepare an SLN and NLC of your topical drug with the size range between 20 to 200 nanometer, it will not rupture the lipid bilayer. Most of the drugs, which are in that range, will enter. So which will give better permeation. Even SLN and NLC will use in ophthalmic drug delivery by enhancing the ocular bioavailability. Why? Because it will be because of the adhesive nature, it will be available in the cornea for a long time. So the drainage of even if the normal eye drops will get drained quickly, these particles will adhere into the corneal region, and sometimes it will enter into the inside of the cornea by using some modification. Surface modification also is possible. Okay, so thereby it increases the things. Okay, ocular bioavailability. And this is our recent publication of nanodrug delivery, how it will useful for CNS drug delivery. So coming to the conclusion, so it's a frontier area. Solid lipid nanoparticles, or NLC, is a frontier area for future scientific and technological development, and it's one of the promising drug delivery system which can be a potential use in controlling as well as targeting the drug delivery. And SLNs are used for parenteral, oral, ocular, and transdermal route as well as used as a sustained release dosage form. So SLN and NLCs for drug delivery and targeting will have very mature, faster, and become more useful for many lesser therapeutic drugs, especially class 2 and class 4 drugs. With this, I'm concluding my presentation, and I would like to thank my university, JSS Academy of Higher Education Research, Mysuru, and my principal faculty members, students of JSS College of Pharmacy, Ooty, and my sincere thanks to the management and my brother, Mr. Dr. Ramkant, who has given the opportunity to share my experience of SLN and NLC. I mean, because I worked in this area from 2010. I mean, almost like 10 years of experience I'm having in this area. So it's a nice opportunity or platform for me to share my knowledge to the needy people. So thank you once again for the patient listening. Thank you, one and all.
Thank you, sir. Thank you so much for your valuable and wonderful session. So now it's time for the participants to ask any suggestions or doubts to the speaker so that you can get clarified.
Definitely.
Or else you can post.
Participants can unmute your phone and you can post your questions.
Sir, speaker sir, one question is there. How zeta potential will affect nanoparticle stability? Is there any standard values?
Yeah, it's a good question. Already I have discussed, but still I'm emphasizing once again. I told you, the stability of the particles supposed to be plus or minus 60 is the most standard value which confirms the Pakka stability of nanoparticles. But achieving that millivolt potential of zeta potential is very difficult. So we will keep the range between plus or minus 30. That is a desirable range. That's a standard range. Actually, in many papers you might be knowing, or you might come across that plus or minus 30 millivolt of zeta potential is fine. Even plus or minus 20 millivolt is desirable. So below this or above that will not have much stability. So if the particles having this surface charge, means obviously it will not go for any kind of agglomeration. It will be an intact particle. So thereby we can assure or assume that those particles are more stable than that of other zeta potential. That's an answer for the particular question. Hope I am justified.
Thank you, sir, for the answer. So, any other question?
Yes, sir.
Yes.
This is Sindil Kumar.
Yes, sir.
How far this PDA is going to be influencing for biological activities, sir? PDA,
Yeah,
polydisperse index.
Yeah,
Whether it is going to be vary for oral route or topical route. Is there anything will be there for the differentiation? How we are going to be differentiated, sir?
Yeah, that's a good question. Thank you, doctor. And I already told PDA. If the PDA is very close to 0, means I said that's called unimodal particles. Okay. So obviously unimodal particles will have the similar size. Similar surface area also. And accordingly, when you incorporate the drug, the drug loading, will be uniformly, it will entrap the drug. So thereby, when you are fixing the dose, when you are calculating the dose, it will be much much superior if the particles are following unimodal, means your dose calculation and all will be accurate. If at all, if it is showing polymodal close to 1, means for example, if you are what is called calculating particles 1 mg contains this much drug. So, the next 1 mg, it's assumed. For example, 100 mg, we will not what is called calculate how much amount of the drug. So we will take generally some part and we will go for dilution and we will calculate. So if it is unimodal means almost the results will resemble similarly. If it is a polymodal means there is a possibility. The first 1 mg it will show this much amount of the drug. If you take the second 1 mg of your nanoparticles and check the drug, it will vary because irregular size. Irregular size may accommodate different amount of drugs. So that's the reason I can able to justify. So we all know generally uniform in size will be always advantage in terms of anything, not only for the route of administration or therapeutic efficacy. Even in the drug release also, it will be what is called release the drug in a controlled manner. If it is unimodal means, if it is polymodal means, there will be a chance of a sudden burst effect. Even usually the drug release of nanoparticles will be initially burst effect followed by control release. The reason. First, the adsorbed particles on the surface will get released as a burst effect and followed by. But if the particles vary in size, accordingly, sudden changes. I mean, some sort of what I can say, fluctuations in the drug release. So obviously that and all will be a matter in efficacy also.
Thanks a lot, sir.
Yeah.
Thank you, sir.
Is there any standard
one more-
values is there, sir? Any standard values for PDA?
That's what it's supposed to be 0 to 1.
0.
Most
Okay,
of the 0 to 1 as per the Malvern zeta sizer. You can get this value. If you use some other zeta sizer, it will be in percentage. So if lesser the percentage, better your what is called. I mean, uniformity. For example, here we are using something. 10% means 10% of the particles are vary in size. 90% of the particles are the different in size. Some sort of different interpretations are there with different types of, the instrument what we are using. What I am telling is, as far as Malvern zeta sizer, the standard scale range is 0 to 1. If your PDA is close to 0, then most of your particles are unimodal. I mean, it follows unimodal, uniform in size. If your values are close to 1, means your particles are following polymodal, which are having different size.
Thank you, sir. Thanks a lot.
Thank you, Speaker, sir. One more question from Mr. Prakash. So is there any success stories with polysaccharide nanoparticles, and which method is preferred for a polysaccharide as nanoparticle?
Polysaccharide, in the sense, it's a very wide term. Generally, chitosan and all nowadays people are using, and there are so many successful stories are there. Technique, what generally people are using for chitosan based is this. That's what. So I'm not able to remember right now. And there are many people are working with polysaccharides, especially chitosan. People can find many details from Google or something. I think so. I'm not very much sure because I'm rarely working with polysaccharides. Most of my research works are based on the lipid based. If they're asking any questions related to that, I can able to justify. If otherwise, I'm very sorry for that.
Thank you, sir. Thank you. So, participants, again, is there any questions, or you can post it?
Bendik sir, shall I introduce? Yeah. Jawahar sir, thanks a lot for your valuable speech, and the feedback from the participants during the post chat box was very excellent. And on behalf of organizing committee, I would like to highlight a few things about this webinar. The session was very interactive. You made the session highly interactive, and there was totally 239 registration, and of it, the participants were from a wide range of all over world country, and even from AP, Assam, Bihar, Chhattisgarh, Haryana, Indonesia, and Jharkhand, Karnataka, Kerala, Maharashtra, Odisha, Punjab, Rajasthan, Telangana, UP, and so a wide range of West Bengal also even. So apart from the Tamil Nadu local participants. So these are the various participants. Overall, 239 participants. Hope all the audience might be got a good session of it. Thank you, and Pendix, let us move on to the next session.
Thank you, Ramkant. I think I got one question from the chat box. So
One more
I think Dr. Sendil asked about the toxikinetic of nanoparticles. Yeah, that's a big area actually. But as far as lipid-based nanoparticles is concerned, that's what I don't know about the other polymeric nanoparticles which are supposed to be studied. But as far as lipids, lipid-based nanoparticles is concerned, the cytotoxicity is not yet reported much in many of the research papers, as well as even I have gone through the review papers, which states that so far no toxic related issues, or even they performed the toxic studies, nothing has been what is called reported as toxic with respect to lipid-based nanoparticles. So that's the answer for Dr. Senthil. So if we want to go for toxic kinetic studies, we can go for, but as far as lipid-based is concerned, it is not required, since it is used with physiologically accepted lipids, and most of the excipients are also used under the category of GRAS. So we no need to go for toxicokinetic unless until if required.
Thank you, sir.
Yes, sir. To the speaker, to the speaker, one thing is there are several chat box. There was the audience are requesting your PPT, sir. If your permission, if you wish, we may share it. It's up to you, sir.
Jawahar? I will what is called mail to you in the form of PDF so that we can share. Yeah.
So nice of you. Thank you, doctor. Thank you.
Okay then. Thank you.
Bendik sir, we may proceed.
Ramkant sir, shall we conclude with vote of thanks then?
Yes, sir. Please. Yes, sir. Please.
Yes, Ms. Harita.
Yes, sir.
Yeah, you can proceed with vote of thanks.
Okay, sir. Good afternoon to one and all. On behalf of Karpam College of Pharmacy and its entire fraternity and the organizing committee, I feel immense pleasure to propose vote of thanks. Once a great man whispered, feeling thankfulness and not expressing it is wrapping a gift and not giving it. Today, I take this opportunity to express all my gratitude into words. First of all, I would like to thank our distinguished speaker, Dr. N. Jawahar, Associate Professor, Department of Pharmaceutics, JSS College of Pharmacy, Ooty, for gracing today's webinar. Thank you, sir, for your interesting and thought-provoking address. And I'm sure the points about the SLN, modified lipid nanoparticles, its method of preparation, and about the lymphatic drug delivery system was an eye-opening for our delegates. Then I extend my gratitude towards the management of Karpam institutions for the encouragement and support. And also, I wish to express my gratitude to our principal, Dr. S. Mohan, Karpam College of Pharmacy. Thank you, sir, for your motivation and guidance. Next, I would like to thank Dr. S. Ramkant, HOD, Department of Pharmaceutics, Karpam College of Pharmacy. Thank you, sir, for initiating and organizing such an informative session. Then my special thanks to the organizing committee members, all the faculties for their support and coordination. Then my deep sincere thanks to the delegates for the active participation and cooperation throughout the session. Thank you once again. Thank you all.
Thank you, Harita, for your wonderful vote of thanks. And Ramkant sir?
Yes, sir. You may end the session, sir. That's it.
Yes, sir.
So thank you, Doctor. Yeah. Please.
Proceed, sir. Proceed.
Thank you, Doctor, sir, from special from my side. Thank you, sir. Thank you.
Thank you. I'm leaving the meeting. Okay, fine.
Thank you, sir. And thank you all the delegates. So this is the end of the session. Thank you all.